Hi, my name is Samantha Courtney, and I am a post-bachelor student working for the Chemistry for Biomedical Applications team within CPCS. Today, I will be talking to you all about our work on influenza diagnostics that incorporates molecular beacons and a waveguide-based optical biosensor. Let's get started. I first want to give a little background on the influenza virus. Viruses are basically little pods of genetic material and proteins, and as you can see in this figure, influenza is no different. The influenza virion consists of important proteins including hemagglutinin, neuraminidase, the M2 proton channel, as well as 8 separate segments of single-stranded RNA as its genetic material. Each of these components allow the influenza particle to be the extremely infectious particle we all know it to be. Several of the RNA segments have a high mutation rate and have intraspecies transmission and genetic crossover capabilities. With the perfect recipe of virulent factors during crossover, influenza has the potential for a pandemic, and this was proven true in both the 1918 Spanish flu and the 2009 H1N1. Because of this, there is a very large incentive across the globe to have effective preventative, diagnostic, surveillance, and treatment measures. In terms of diagnostics, there are several approaches. Hemagglutinin and neuraminidase are both highly expressed surface glycoproteins, making them the most common biomarkers for protein-based or antigen diagnostics. The most commonly used diagnostic in clinical settings are rapid influenza diagnostic tests. Antibodies are embedded on a nitrocellulose membrane, and as the patient sample, which if positive for influenza should contain the antigen hemagglutinin or neuraminidase, will flow across the membrane and 2 red lines will appear. If there is no target antigen present, only the control line will appear. This test is rapid and inexpensive, but unfortunately there is a high rate of false negatives. Nucleic acid testing is much more reliable, but is not cheap or quick. In real-time polymerase chain reaction, a patient sample is stripped away of everything but the genetic content. If influenza RNA is present, it will be replicated and amplified over and over again until it is at a detectable level, designated by a fluorescent signal. The sensitivity to influenza RNA is almost 99% for most clinically used RT-PCR platforms, and it can also subtype what strain of influenza was present for epidemiological surveillance. Lastly, we have newer methods that require expensive machinery, such as surface-enhanced Raman spectroscopy assays. But the methods being developed are extremely rapid and ultra-sensitive, having results in minutes with information about the specific strain of influenza present. Molecular beacons are common as a fluorescent marker in real-time PCR, but they have been developed as their own nucleic acid diagnostic method without amplification of the genetic material. The structure of a molecular beacon includes a loop sequence, which is designed to complementary bind to a target influenza gene sequence present in all influenza RNA, a stem sequence to stabilize the loop, a fluorophore to indicate a positive sample, and a quencher to dull fluorescence when the beacon is not bound to anything. The beacon has 2 conformations, open and closed. When the beacon is closed, it is not bound to a target sequence of influenza RNA, and very little fluorescence is emitted. When it is open, it is hybridized to a target sequence. The quencher and fluorophore are separated, and fluorescence is emitted. Therefore, a positive sample is detected when the beacon binds to its target. This beacon-to-target hybridization is extremely sensitive, but we need to achieve a low background fluorescence level when the beacon is closed, so that the signal is interpretable when the beacon is open. Our approach takes the concepts of molecular beacons and applies them to our team's waveguide-based optical biosensor. I will delve into each of these parts thoroughly, but first I'll give a quick overview. Our theory team designed an algorithm called Fast Evaluation of Emerging Risks, or FEVER, that aligned every known sequence of influenza A and B and gleaned several molecular beacons that contained consensus sequences of a well-conserved influenza gene. After the design, we had to characterize the thermodynamics of the molecular beacons to determine the optimal binding conditions. We did this by designing synthetic DNA and RNA targets to control and test the beacon-target interactions in order to create straightforward protocol for sample prep and detection guidelines. Lastly, we applied the molecular beacons and synthetic targets on our biosensor platform, hoping for a low, clinically relevant limit of detection. The fever algorithm is a key component to our project. To make successful molecular beacons for influenza detection, the sequence of nucleic acids in the loop sequence of the molecular beacon needed to have a sensitive, specific consensus of a well-conserved gene that does not mutate often. We chose the polymerase acidic gene and chose 3 sequences of nucleic acids that were present in almost every strain of influenza A and B. From there, 3 molecular beacons were designed, 1 for influenza A and 2 for influenza B. Together in a multiplexed assay, they would be able to detect and subtype the influenza sample. We then designed synthetic DNA and RNA targets that were either a complete complementary match to the beacon or contained 1 to 2 mismatches. The mismatched targets were tested to simulate a more realistic scenario, in which a patient's influenza sample would predictably have a few mutated nucleotides in the PA gene. Our hypothesis was that the beacon would bind tightly and emit fluorescence for both the complementary match and the mismatched target. To test our hypothesis, we first characterized the molecular beacon interactions with the synthetic targets in a controlled environment using a melt curve experiment, which ramps up the temperature at 1 degree increments from room temperature to 95 degrees Celsius. DNA is double-stranded and temperature-dependent, so at room temperature the 2 strands are bound, and at high temperatures the strands spontaneously dissociate into separated single strands. Likewise, a single-stranded synthetic target and a single-stranded molecular beacon bind together at room temperature and spontaneously dissociate at high temperatures. We can take advantage of these binding properties and measure the fluorescent signal at room temperature as a signal that the molecular beacon is open and bound to the target. Then, when they dissociate, the beacon closes and returns to its base level of fluorescence. As you can see on the right-hand side, these graphs look almost identical. Regardless of the concentration of the target, there was a strong fluorescent signal at room temp and a weaker signal at high temps for both the matched target and mismatched target. This confirmed that the molecular beacon behaved properly and that it tolerated a few mismatched bases. This gave us confidence to move on to our biosensor application. Our waveguide-based optical biosensor was originally designed for bacterial diagnostics, but our project was the first to employ viral diagnostics on the sensor. To make our molecular beacons compatible with the sensor, we conjugated the beacon to biotin, which would not interrupt the beacon's binding mechanism to the synthetic target. The surface of the waveguide is coated with streptavidin, and because we know the streptavidin biotin proteins tightly bind together, we can ensure that the beacon is anchored to the waveguide surface for analysis. Because fiber optic waveguides are made up of a material with a high refractive index, coupled light can propagate through the waveguide to generate an electromagnetic wave and excite the molecular beacon fluorophores for ultrasensitive detection. So, when the molecular beacon binds to the target, the fluorescent signal will be detected immediately by the sensor. The figure on the right confirms that, compared to the thermocycler in which the melt curve characterization experiments were performed, the sensor had a much higher signal-to-background ratio, meaning the beacon's fluorescent signal was very clear and very strong on the sensor. Using the RNA synthetic target on the sensor, we detected 10 to the 13th RNA strands per microliter. Unfortunately, this was not as sensitive as we were hoping. It does not make our diagnostic platform competitive in the field of influenza diagnostics yet. Presently, we are continuing to improve the limit of detection on our sensor, and we aim to perform cross-reactivity experiments to make our data experimentally relevant. However, we do want to attempt to apply our probes to other diagnostic platforms such as flow cytometry or SPR, so that 1, all our eggs aren't in one basket, and 2, to see if our beacons are more compatible on another diagnostic platform to achieve a lower limit of detection. Overall, our future studies will maintain the goals of producing a diagnostic method that reduces performance time, increases sensitivity and specificity, and detects the influenza while simultaneously producing immediate data to further epidemiological surveillance of influenza. I'd like to thank my amazing fever team, which comprises of members from both T6 and CPCS. That is all I have for you today. Thank you for your time.